So now I think we can move on to our guest speaker, and it gives me a great privilege, and I'm very happy to call upon Dr. Rob DeSalle for his presentation.
Buenos dias, and gracias to Robbie Burk and the organizers of this conference for inviting me. It's a real honor for me to be here. I have to confess to feeling a lot like a fish out of water, because I usually spend my time hidden away in the collections at a museum. And you might be wondering, well, why is a guy like that here giving a talk at a medically important conference? Well, it turns out that the study of evolution and the application of evolution to how viruses evolve and how viruses interact with us is quite important. So the title of my talk, as Robbie mentioned, just added HPV at the beginning. So HPV, Human Genomes and Human Evolution. And I subtitled it, Nothing in HPV, Human Biology Makes Sense Except in the Light of Evolution. And this is a ripoff of a famous quote from Theodosius Dobzhansky, who was a famous evolutionary biologist. It's quite a good quote, and it's quite a good start for this talk. Evolution is very, very important for how we understand this virus and how it actually interacts with us. And what I want to do today is I want to talk about three things. First of all, what did Darwin do right? Darwin is the originator of evolutionary thought, really. What did he do right, and how does that help us understand HPV? And then so I want to talk about the evolutionary history of viruses in humans and that relevance to understanding HPV. And then finally, what do human genomes tell us that is relevant to HPV biology? Unless these HPV viruses are agnostic to human genomes and what human genomes are doing, then this last thing isn't that important. But I doubt very much that these viruses are agnostic or unresponsive to the things that are going on in our genomes. And so what I hope to do is give you kind of a background for evolution and how the human genome has to be looked at when we start to think about HPV. So first of all, what did Darwin do right, and how does that relate to HPV? Well, the first thing that Darwin did right was he presented overwhelming evidence for the existence of evolution. And no one had done this before. Evolution was an idea that was out there prior to Darwin, but what he did was he presented overwhelming evidence. In fact, he called his On the Origin of Species one long argument. And he also understood something that's really, really critical to the understanding of HPV and human evolution, and that is coevolution. And even though he didn't coin the term, the term wasn't coined until the 20th century, he absolutely understood that organisms interacted with each other in order to produce the evolutionary product. And obviously he didn't know what viruses were, but he had a great idea as to how things would co-evolve with each other. He called it co-adaptation. And in this slide I show his famous orchid example and his equally famous book on the contrivances. The second thing that he did was he provided a mechanism. And this mechanism is natural selection, and that was absolutely unique in the history of biology. No one had given this mechanism or thought of a mechanism for how evolution would occur except for wrong mechanisms, as Lamarck had developed. And by focusing on variation and trying to get natural historians to understand variation, what he did was discover and articulate a new force that existed on our planet. Really, really very important. And the force is now one of the cornerstones of modern biology. And the famous example that he that he came up with during his studies and during the voyage of the Beagle were these Darwin's finches, in which he showed case after case of how natural selection worked in those organisms. And now we have textbook examples of natural selection. HIV and antiviral treatment is an absolute textbook example now in high school textbooks and in college textbooks. And not only do we have that, but we also have a modern formulation of natural selection at the sequence level. And in this slide we see the 4 codons for the amino acid proline. And proline is an important amino acid in our proteins. At the DNA sequence level, it's coded for by 4 different codons. That last position is called a silent position, And the two previous positions are called replacement positions. And under a hypothesis of evolution just occurring neutrally, you'd expect equal numbers of those kinds of substitutions. And what we can do nowadays is we can count the number of changes that occur in silent positions and replacement positions and come up with a kind of a lexicon for how natural selection is working at the sequence level. In fact, if we count many, many more replacement substitutions, i.e. substitutions in those first two positions relative to replacements in the last position, then we know natural selection is actually working on that protein and forcing that protein or pushing that protein in a particular direction of evolutionary change. So the last thing that he did was he changed the way we think. And a lot of people don't think of Darwin as a philosopher, but he indeed changed the way that we think and the way that we philosophize about nature around us. And in this way, as I mentioned earlier, he interested us in variation. And variation was something that people tried to ignore up until his time. In fact, people thought about the natural world in a very typological way. That is, they tried to look for the means and for the types that help them explain the natural world. And what Darwin said is, hey, wait a minute, it's the variation that's really important. And so this focus on variation becomes very, very important for not only the way Darwin articulated evolution, but for how we today understand how evolution proceeds. And he also came up with this other really neat thing called tree thinking. He called it the great tree of life. And On the Origin of Species, this is a quote from On the Origin of Species, and he suggests that there is a great tree, and we oftentimes represent life on this planet by this great tree. And he thinks that the similarity is very, very true. And the figure that you see here, it's a very, very washed out figure because it's the only figure from On the Origin of Species, from the first edition of On the Origin of Species. The only figure in his book was a tree, and introduced his way of thinking about the natural world in a tree thinking way. And in a letter to his friend J.D. Hooker, he said, I discussed the principle of divergence, which is tree thinking, which along with natural selection is the keystone of my book. So he recognized that tree thinking was also an important part of how we would understand evolution. And this is his famous, I think, figure from his notebooks in the 1830s and 1840s. And in this figure, he discusses how the relationships of B and D in the figure are closer than the relationships of B to A. This is a really, really exquisite understanding of common ancestry and descent with modification. And it's such a prevalent idea that a lot of graduate students and postdocs have taken to tattooing themselves with this iconic understanding of the natural world, that is, this tree thinking way of understanding the natural world. And so these are the three things that he did right, and I hope this sets the stage for how we're going to think about HPV and the human genome and human evolution in the next few minutes. So let's think about the evolutionary history that we see on this planet, this so-called great tree of life, and its relevance to understanding HPV. And we can ask three things about the tree of life. We're a very self-centric species. And we always have to ask, where are we? You know, we always need to know where we are. The tree of life is actually really, really an interesting way to think about where we are. We can also ask, where are viruses? In fact, are viruses really part of the tree of life? And then finally, what about the history of life can we say about HPV and us? And this is the homepage from a website called The Tree of Life. It's a project that's been ongoing for the last 25 years. Most of you may not know about it because it's been done by museum scientists, and we like to hide, and so this project has been hidden away. But this is a treasure trove of information about relationships of organisms on the planet. And this Tree of Life can be translated into all kinds of forms, such as this circle tree that you see here. In this tree, the green organisms are archaea, the red organisms are eukarya, and the blue organisms are bacteria. The tree of life, if we drew it proportional to all of the organisms on the planet, would have much, much more blue in it and much, much more green in it and very little red in it, really. Eukaryotes are the minority party, so to speak, in the tree of life. And that red dot in this tree of life that we see on the slide, in this highly reduced tree of life, this is only about 200 species out of the 1.7 million known living species. That red dot represents where Homo sapiens is. we can focus in on Homo sapiens a little bit better. And this is work from Perry et al. That shows using 7,000 or so genes, the relationships of primates, of some of the primates. There are about 300 species of primates, and we're only showing 18 here. But you can see humans are at the very top of the tree, and they show close relationship to chimpanzee. And then we have all of these other really interesting primates at the base of the tree of life, of the primate tree of life. And this is our family tree, really. This is focusing in really, really closely on our family tree, and these 5 beautiful females that I show you on this slide represent the 5 major higher primates: the gibbons on the left, orangutan, gorilla, the common chimp, bonobo, and my wife, Erin. She loves this slide. The timing of how this occurred, too, is very, very interesting. So the whole group of higher primates probably arose about 17 million years ago. The orangutan had a common ancestor with us. Orangutan lineage had a common ancestor with us about 12 million years ago. And 9 million years ago, the gorilla lineage broke off from chimpanzees and us. This is very well understood now from molecular data. When I was a graduate student, we used to call the gorilla-chimp-human relationship the holy trichotomy. It hadn't been worked out 25, 30 years ago, but now there's overwhelming evidence from DNA sequences and behavior and anatomy now that I think unequivocally states that chimpanzees are our closest relative. Now, there are two kinds of chimpanzees, bonobos and the common chimps, and they diverged from each other about 2.5 million years ago. And these two kinds of chimps actually are very, very different behaviorally. The common chimps are nasty, nasty animals, as those of you who use them as a model system, perhaps for studying HPV, will know. The bonobos are very, very mellow individuals, and in fact, the common chimps solve conflict using violence, and bonobos solve conflict using sex. Not unlike probably a mixture of the human lineage, where we tend to solve conflicts with both of those ways. Now, you'll notice that on this lineage leading to humans, it's kind of naked. What is happening on this lineage? Well, it turns out that a lot was happening there. There's not just a single species that diverged from our common ancestor with chimpanzees and bonobos. There were 24 or so, there are 24 known species of humans that existed on the planet along that 6-7 million year branch. And one really interesting thing, if you look at the kind of the middle of the diagram at around 2 million years, the diagram shows present time at the top, and then the first hash mark is 1 million years ago, the second hash mark is 2. At around 2 million years ago, there were 7 species of humans walking the planet. This is phenomenal. And what this means is that there were lots of highly diverged lineages of people walking around the planet. And this history goes all the way back, like I say, to about 7 million years ago, and it's based entirely on fossil evidence. And as we'll see, we do have some genome sequences of fossils, but for the most part, we base this set of relationships on the fossils. And again, what this tells us is that there are a lot of differentiated lineages of humans out there. And even up until about 30,000 years ago, there were 3 species of humans walking the planet. Homo neanderthalensis, who we'll talk about here in a second, Homo sapiens, us, and Homo floresiensis. This is a small species of humans who lived on the island of Flores in Southeast Asia. And there's probably yet another species around 30,000 years ago that's related to a specific fossil called Denisova, which I'll talk about in a second. So the last thing I wanted to point out about our existence is that it's minuscule, it's fleeting, it's so tiny with respect to the history of the universe. And this is I think best shown by an exhibit at the American Museum of Natural History in our planetarium. It's called the Walk of Time. And you walk into it and you start at the Big Bang, which was about 13 billion years ago. And one step equals about 75 million years. So as you're walking through time. And it's not until you get about halfway through the walk that the Earth forms. And it's not until you get about 3/4 of the way through the walk that life forms on the planet. And it's not until you get about 10 feet away from the end, 10 or 15 feet away from the end of the exhibit, that complex eukaryotic life evolved on the planet. And that last arrow shows where the dinosaurs went extinct. And at the very end of this exhibit is a human hair. And this human hair represents the amount of time that Homo sapiens has been senescent. That is, it represents the amount of time that we have been classified as modern humans, only about 180,000 years. So our existence as a species, as Homo sapiens, is very, very small. And indeed, as I'll show you here in a second, our existence as a species moving out of our home continent, Africa, is even shorter, is even smaller. So there are three major ideas as to how humans evolved on this planet. One is the candelabra idea, and this is the idea that all of the different continental groups of people arose as a burst. There's also the multi-regional hypothesis, where this burst occurred and that was followed by interaction and interbreeding of populations. And then there's the replacement idea that basically comes from Alan Templeton and other human geneticists. And this replacement idea is perhaps the best way of thinking about how we evolved, and that is because of the incredibly narrow amount of genetic variation that we have amongst all of the living people on this planet. And in fact, this has led us to think of our evolution as starting about 180,000 years or so ago in Central Africa. Lineages moved around Africa and formed in Africa for the next 120,000 years. And then about 50,000 to 60,000 years ago, there were massive migrations from Africa into Asia, Australasia, Australia, and Europe. And then a final migration about 12 to 17,000 years ago into the New World, into North and South America. And this brings us back to another way of thinking about humans and how humans have evolved on this planet, and that is, what do fossil genomes tell us? I'm kind of getting ahead of myself because I want to talk in detail about human genomes, but fossil genomes at this point in time are important. And we can get whole fossil genomes now. This is because of the amazing technology developed by my friend and colleague Svante Pääbo at the Max Planck Institute in Germany. Amazing technology whereby DNA is isolated from bone shards or from teeth, and the DNA is then sequenced using high throughput next generation sequencing to give us high coverage sequences of these ancient genomes. The individual on the left is a Denisovan female reconstructed from DNA made from a finger bone, and the individual on the right is a Neanderthal reconstructed from multiple fossils. And both of these genomes tell us one thing that's really, really important, and that is we like to do it. And we like to do it with whatever we can find, really. And what this means is that the fossil genomes tell us that we have interbred with these other lineages. And that's why knowing that we have these other lineages around is really important, because knowing that these other lineages were around and they interbred with us gives us even that much more information about the variation in our populations. And in essence, the replacement hypothesis gets changed into an assimilation hypothesis, whereby Denisovans and Neanderthals are assimilated back into the Homo sapiens lineage. And it's estimated that about 1 to 4% of the genomes of Northern Europeans and some Northern Asians contain Neanderthal specifically Neanderthal sequences or uniquely Neanderthal sequences, and a small proportion of Southeast Asian genomes contain some of the Denisovan, strictly Denisovan kind of DNA. Now, that's humans. So what about viruses? Well, there's a really interesting paper that was published a few years back that was titled 10 Reasons Why Viruses Should Not Be Included in the Tree of Life, one of the most negative papers I've read in a long time. And the two main reasons why viruses should not be included in the Tree of Life are that viruses are polyphyletic. And polyphyletic is just a term that phylogeneticists and systematists use to describe multiple origins. There's no single common ancestor of all viruses. And this is somewhat problematic in trying to understand where viruses came from. They simply don't have a single common origin. They have many separate and disparate evolutionary origins. And the diagram on the left shows a typical virus and the diagram is supposed to represent about 180 or so genes in viruses that are also found in cells. And of those 180 genes, 60 to 70% of those can be shown to be of eukaryote or eukaryote origin. About 30 or so percent can be shown to be of bacterial origin, 1% of archaeal origin, and about 7 or 8% of unknown origin. And only a very small number, say 6 or 7 genes of those 180, are actually unequivocally of viral origin. Now, the other really interesting thing about these genes is that no single gene is found in every virus. And this makes it really difficult, impossible to really discover the common ancestor of all viruses. And so viruses are generally not kept or not put into the tree of life. In fact, this is the shape of a viral tree of life, and the phylogeneticist who sees this will shudder because of a lack of resolution here. While there is some resolution, such as in the lower left-hand corner of the tree, those are relationships amongst really well-known groups of viruses. But as you get further and further away from these well-known groups, you see a complete lack of resolution. And this is highly problematic for trying to understand the origin of viruses. But all is not lost. As Ignacio will tell you, there's a lot of structure within a particular kind of virus. And this is what I call the shape of PV evolution. And this shape of PV evolution can give us a lot of information about the origin of viruses and the origin of their coevolution with us. And even within a separate kind of virus, such as HPV-16, there's a lot of resolution, so all is not lost. Now, we also know that microbes oftentimes mirror human geography. And in fact, this example on the left from Helicobacter from Martin Blazer's lab at NYU is a classic example of microbes actually mirroring how we moved across the planet. And on the right is another example from my colleague, Paige Caulfield at NYU, who is convinced that Streptococcus also follows the geographic movement of people across the planet. And then finally, this HPV-16 example from Robbie Burk's lab, it demonstrates nicely how the virus can follow the movement of human populations. And since these microbes mirror these human geographies, we need to understand the phylogenetics of these viruses and the phylogenetics of these human populations, of which understanding human population phylogenetics is very, very difficult, as I hope to show you here in a second. So finally, what I want to do is to ask, what do human genomes tell us that is relevant to HPV biology? And again, unless HPV is agnostic to the genomes of humans and is agnostic to how the genomes of humans produce proteins and how the genes are expressed, then we need not talk about this. But it's very important at this point to point out that that HPV is probably not at all agnostic to our genome, and that our genomes have co-evolved with the HPV genome. So the problem becomes one of how do you understand the relationships of people on this planet. And this beautiful statue from the Musée d'Orsay shows 4 women of different ethnic backgrounds holding up the world. And the modern day version of this is this slide showing Shaquille O'Neal, Yao Ming, and Christina Aguilera in the same picture, demonstrating the breadth of variation across human populations. And while we know that humans are very, very different when we look at them, we still need to understand how we're all related to each other, if indeed there is a way to understand how we're related to each other. And so lots of human genomes have been sequenced. First off, the 2 very, very famous scientists' genome, and next a 3rd famous scientist's genome, some very important African sequences, less scientists who are very, very involved in the sequencing technology were sequenced next, and then, of course, you get businessmen who have a lot of money that they can throw away to have their genome sequenced. Those are the last 2 guys that showed up. And then the very last 2 guys that showed up, Neanderthal and the Denisovan female, have been sequenced too. Now, these individual genomes tell us a lot about the degree of variation amongst human genomes. And in fact, the whole of human variation has been approached in a very systematic way now with the 1,000 Genomes Project, which probably should have been called the 2,500 Genomes Project, because instead of sequencing 1,000 genomes at high coverage, these guys are sequencing 2,500 at high coverage. And this gives us a deep catalog of human genetic variation, but even more recently with the ability for geneticists to write prescriptions for whole genome sequences, the number of genomes has exploded to over 10,000 to 15,000. So there's a huge number of whole genome sequences out there that are giving us a treasure trove of information about the human genome and how these genomes interact with all kinds of diseases and all kinds of environments. And this is a list of the different kinds of humans that have been sequenced. The sequencing is done using subscribed groups, and within those groups, oftentimes a parent-offspring trio is sequenced. And these groups are then used in genetic studies that then try to focus on the origin of disease and the origin of genetic disorders. So what can these human genomes tell us about ourselves? Well, first of all, these human genomes can be used to understand natural selection and how natural selection is working at the genome level using that silent replacement approach that I mentioned in the first few slides. It can also tell us something about our relationships, but as I hope to point out, we have to be very, very careful about how we think about that. And then finally, it can tell us something about how the variation is apportioned amongst human populations. And this variation becomes very, very important when we try to understand how the human genome is confronting challenges like HPV or Streptococcus or Helicobacter. Now, the human genome is categorized into single nucleotide polymorphisms and their position. And these positions are known with respect to a reference sequence in the human genome, and these positions are categorized as encoding regions or not encoding regions, and in silent and not in silent sites, and so on. And this slide just shows a single nucleotide polymorphism. The red G is polymorphic, and this would become a SNP that is now categorized and stored in a SNP database. And when the human genomes were first sequenced, it was shown that the average number of single nucleotide polymorphisms between any two randomly chosen individuals was about 0.1% of the entire genome. And with only a few genomes sequenced, say, 6 or 7 years ago, this meant that there were a few million SNPs that had been discovered. Subsequent studies have increased the number of SNPs to over 20 million, and many of these SNPs are what we call rare, in that they appear in the genomes of only one or a few genomes so far sequenced. So we have to distinguish between these rare variants that can tell us certain things and these more common variants that are probably more important for understanding the genetic basis of disease. And just recently, a really wonderful study from Sarah Tishkoff's lab at the University of Pennsylvania came out where they sequenced 5 individuals from 3 very old African populations. And just to give you an idea as to the kind of variation that's found in these African populations, they found 3 to 5 million new SNPs. This is, they increased the number of SNPs by about 30 to 40% simply by understanding the genetics of the genomics of these African populations. And they use very high resolution sequencing to do this. Now, what you can do with respect to natural selection is you can scan each one of these SNPs and ask the SNPs, or ask a stretch of SNPs or a window of SNPs that are contiguous to each other, whether or not natural selection is occurring in those regions. And you can find regions, as shown with the dots down at the bottom, you can find regions that at different genomic positions that far exceed the levels of selection or far exceed some proxy for the level of selection. And these regions then become candidate regions for SNPs or SNPs, and potentially for genes that are involved in interacting with the environment via natural selection. As far as relationships, and this is something that I wanted to address because it's very, very important, as far as relationships, we have to worry about the markers that we're using when we talk about relationships. And a lot of the relationships that I've talked about so far with respect to the relationships of human populations are based on clonal markers, that is, on mitochondrial DNA, which is inherited from mother to offspring, and Y chromosomal DNA, which is inherited from father to son. And if you look at common SNP variants and ask, what does a Y chromosomal set of relationships look like, you get this. And unlike that viral tree that I showed you a little bit earlier, this is a little bit more pleasing to a phylogeneticist because of the degree of resolution. But if you go and you look at a haplodiploid part of the genome, the X chromosome, you start to see an erosion of variation until you go to a diploid chromosome and you see a complete erosion of resolution, almost a complete erosion of resolution. In fact, the only things that are being resolved here are familial relationships. This doesn't mean that individual genes on chromosome 20 will give us a pattern of descent with modification, but overall, the whole chromosome itself is not going to give us a resolved idea of how people are related to each other. And this, again, is because of the non-clonal nature of an autosomal chromosome. And the other thing I wanted to point out is that the kinds of variants that you use, that is the degree with which a variant varies, is important. In this slide I show for the Y chromosome variants that are found in at least 10 individuals on the far left, variants that are found in at least 5 individuals, and extremely rare variants such as on the right. And as you use more and more rare variants, you get more and more resolution when you're talking about clonal systems. But when you're looking at the X chromosome, that variation, increasing the variation doesn't help. And in fact, for the chromosome 20, the resolution of relationships of people is again at the tips and more than likely resolution at the familial level. So this increase in the kind of variation that you're using to do these trees doesn't really help the story much at all. So the final thing I want to address is how variation can be looked at in humans. And the way I want to do this is I want to ask two questions relevant to what do Stephen Colbert and Charles Darwin have in common, other than being from Northern European descent. What do they have in common? And this leads us to two questions. The first is, would Charles Darwin pay for a DNA ancestry test? And the second is, Stephen Colbert really, as he puts it, the inescapable black hole of white people. So Charles Darwin was very, very cognizant of genealogy, as that one figure from On the Origin of Species shows. And this is his pedigree, going back four to 6 generations in time and forward 4 to 6 generations. And the blue simply marks his paternal inheritance, and the red marks his maternal inheritance. And in fact, Darwin, none of his sisters reproduced, and he had 4 daughters, and none of them reproduced. His maternal inheritance is kind of stopped with him. And the idea here is to show what the trees of these two markers would look like. And indeed, if we were able to go back and sequence Y chromosomes and mitochondrial DNA of these individuals, we would see these two tree topologies. And they're very different. And what this means is that different markers tell different evolutionary stories. And Darwin, I think, would have recognized that. And this is something that we have to keep in mind when we think about human populations and human relationships. Now, to get to the Stephen Colbert question, we need to talk about aims. And aims are ancestral informative markers. And these are cherry-picked markers. And what you do is you cherry-pick these markers and use the all swans are white approach. That is, you find markers that look like they're specific to Caucasians, and then you call those Caucasian markers. And these aims have led Skip Gates, a famous social scientist at Harvard University, to use this technique to describe the ancestry of people. And on his show on PBS, Faces of America, he presents many people with their ancestry records. And to Stephen Colbert, he presented a all Caucasian record, and to Yo-Yo Ma and all Asian record, and to Eva Longoria, a mixture of origins for her. Now, this prompted Stephen Colbert to claim that he was the inescapable black hole of all white people. And Skip Gates called him the whitest person he'd ever met. So you can really be confused by this, because in actuality, those cherry-picked markers are only a few hundred to 1,000 in number. You never really know, because the companies that do this use these as proprietary information. But if you consider all the SNPs in the genome, this is what it looks like. Stephen Colbert is in fact mostly almost 99% African, 99.9% African. Yo-Yo Ma is 99.9% African, and Eva Longoria is 99.9% African. What we tend to do is we focus on the variation that makes us look different, and not the variation that makes us the same, really. And this is something that we need to keep in mind when we talk about variation and how it's going to impact our understanding of how pathogens interact with us. So finally, we have several challenges. And these challenges are, first of all, that we need to understand human history and viral history, both shallow and deep. And this will develop a better picture of the coevolution of HPV and humans. We need an understanding of the variation in the human genome in the context of interaction with HPV. This will be important in developing a better picture of coevolution HPV in humans. We need to increase the sample size of coupled HPV human genomes. That is, we need to increase the sample size of sequenced HPV genomes and the genomes of the humans that particular HPV infected. And we need to increase the sample size of animal PVs in order to get a better understanding of the deep evolutionary history of the relationship of PVs and HPVs with human populations. So I hope this gives you a pretty interesting and good introduction to how we are going to think about genomes and how this information is going to be really important in the future in understanding this important virus. Thanks.